Thanks for being here. I am Bill Faber, and I teach chemistry here at Grand Rapids Community College. And in the fall of 2020, I went on sabbatical and worked at a lab at MSU, just down the street here. And one of the things I focused on during that time was learning a little bit more about RNA sequencing. So what I thought I'd do is, as a presentation, is I'd walk you through some of these ideas. And my goal is really not necessarily for everybody to have this comprehensive view of what I did, but I think the idea is that you learn a little bit more about one of these components, because it is sort of an overwhelming thing. As I was learning about it, I would record my discussions with folks in the lab so that I could go back and re-listen to them. That's how complicated it was for me to get my head wrapped around this. So really what I want to do is I'm going to give you a little bit of background on RNA. And then I'm going to talk about something called the transcriptome. And I wish I could just talk about the transcriptome, but I don't think it makes sense without a little bit of background. I will tell you, so one of the huge benefits of doing this work was it really expanded my mind on things that from 25 years ago when I was in school, and even following the research on genetics and applications of genetic components, it is much more complicated than I thought. And so I really learned a lot just about what my lack of knowledge was. And that's what my goal of this is, for you to learn a little bit more about one of these components. So after I talk about the transcriptome, I'm going to talk about where do you get samples to actually do some of this work. Because you don't necessarily have to be in a lab to do what I did. There's a lot of information out there, and genetic information has so much, I guess it holds secrets to things, and I'm going to show you where you could get some of that information if you wanted to study it. And then I'm going to talk a little bit about alignments and applications, why we would do this work, and then what am I interested in and continuing to do. Because one of the goals of my sabbatical was to come up with processes that students can do, not only in my classes, but also in a research club. All right? So when you think, I was looking back at a presentation I gave about a year ago, and I had this same slide or a similar slide about the central dogma of genetics. And we all know that when you think of genetics, you think of DNA can make more DNA. That's how our cells replicate. But then the whole point of DNA is that it codes for RNA, and that RNA actually codes for a protein. And if you know anything about protein synthesis, proteins are the things that make chemical reactions happen in our bodies or in any biological system. So oftentimes drugs are designed to interact with proteins to speed them up or slow them down. They also can be now pieces of RNA that regulate whether or not a protein is created or not. And so this is the central dogma of genetics. And what I always think about dogma is dogma can get really complicated. And it can, because that basic idea that we talk about in a biochemistry class or a biology class, that DNA becomes RNA, is we know that RNA can go back to DNA. And if we think of what's on everybody's minds right now about a coronavirus, is that's what they do. They have a piece of mRNA that can generate a piece of DNA. And so this is more cyclical in systems than we used to believe. And we still know that RNA can become protein through working through ribosomes, but the fact that RNA can actually interact with itself is sort of a new idea, because we know that certain cells have certain proteins that are expressed. You wouldn't want liver cells being expressed, or the genes in liver cells being expressed in your eyes. And so we know that genes are regulated. And one of the ways that they get regulated is through this interaction with RNA. And that is one of the things that I'm going to talk about. So it's complicated, all right? And when you think of classics, and anyone in a biology class would look at this and go, oh yeah, I know those. I know what mRNA is. That's the basic, thing that comes off of a piece of DNA. And you might have even heard of rRNA, which is a component of the ribosome, and I'm going to show you a couple pictures of some ribosomes. But ribosomes are the machinery that takes RNA and makes a protein. Okay? And then there's tRNA, which is transfer RNA, and that carries those components of those proteins, those little amino acids, to that ribosome to make a protein. And these are sort of the classics. And I'm going to show you a couple pictures. I'm going to come back to that. But you might have seen cartoons like this where this represents these two proteins actually represent this little blob here and this bigger blob here, represent a ribosome. And those are actually made of both protein and RNA. And these little colorful beads would be the protein that's coming off of them. And this blue line would be a piece of mRNA. And so we know that RNA is involved in the creation of these proteins, okay, that fold up, and I'll give more pictures of those if you struggle with what that is. But you can see here's a piece of mRNA. It sort of holds the code from the DNA, and there's this is actually a piece of RNA here that's carrying an amino acid. And so RNA is really involved in the creation of proteins. Okay, we only think of amino acids sometimes, but there's all these different different types of RNA that are actually involved in this process. And I went in sort of thinking, and I knew there were other types of RNA, but these classics are what we're kind of aware of. But there's also this hnRNA, which are these unprocessed pieces of RNA that we're going to talk a little bit about. There's long non-coding RNA. There's these little snRNAs involved in a concept called splicing that I'm going to cover. There's little pieces of siRNA involved in interference or turning genes on and off. Same with MI or microRNA. There are just so many different ways that RNA is involved in the cell that it would make sense that people study this. And that is what I learned when I was at MSU. So I'm going to focus a little bit on just this hnRNA. Okay? So if you take a piece of DNA and make a copy of it in terms of RNA, you end up with this kind of long piece of RNA in the nucleus, okay? And then it needs to be processed. And the way it's processed is something that we studied in the fall, and it continues to be studied a lot of different places. But there's this idea that it gets spliced, okay? And there are these different pieces on RNA. There's exons and introns and exons and introns, and these aren't to scale or anything. But exons actually code for proteins. So they have genetic information that tells that ribosome how to make a protein. But then there's these long stretches called introns, and if anybody was around when the Human Genome Project got off and started, a lot of people called this junk DNA. And it was areas of DNA that didn't code directly for proteins. And now we know that junk DNA isn't junk. It's extremely important in whether or not genes are turned on and turned off, and how and related to human health. So and one of the things when I was in the middle of all this, I realized is this is getting so complicated. If we have a disease or an illness, we won't even know what's going on with us. And it's just because it can get so nuanced how people get disease. So I am going to talk a little bit about these pieces of exons that exist that ultimately code for proteins. Now I put this up here because this piece of spliced RNA, which ultimately goes and becomes a protein, there are a couple things that are interesting on it. Eukaryotes put this poly-A tail, this long stretch of A's, and when you think of DNA and RNA, A, T, C, and G, these different bases, and so these are on all these pieces of processed RNA, and it's done for a couple reasons. It helps it get out of the nucleus of the cell, and it's also involved in breaking this down over time. But that's actually a really important portion of this story I'm going to tell about how we get this RNA and study it. Okay, so this is an image of a piece of DNA at the top. Okay, and you can still see those exons 1, 2, 3, and 4, and 5, and they have these stretches of introns that are in them as well. Okay, that's those spaces between. Now that gets translated into that in a eukaryote into this piece of RNA, but then it needs to be cut out. Portions of it need to be cut out, and they can be cut out depending on the situation. So you can see that you could splice, it's called splicing, this idea, and they can be spliced into 1, 2, 3, 4, and 5. Maybe all the different exons are pieced together. There can be missing exons that code for different things, and then they can just be in different orders. And you can see that each one, so that one piece, that one stretch of DNA that we used to think codes for a protein, can actually get spliced together and code for, in this case, 3 different proteins. And they may share some components along with them, but they could function differently. They could be regulated differently. There are environmental factors that might have an influence on which ones produce during this. And so this is an important thing to understand as we start talking about the work that I did. So just understand that DNA, a stretch of DNA, can actually code for multiple proteins. Okay? Now, one of the things I put down here is a note that those different proteins have their own mRNA sequence. So you can see this sequence, because it contains different exons than this one, would have a unique sequence. We call that a transcript. Each one of these pieces, this is a transcript for protein A, this is a transcript for protein B, and so on and so forth. Now, the reason I tell you that is because that transcript library is really important to the work that I did. Okay? Now, one of the also that I want to emphasize, because I want to talk about ultimately how you can diagnose disease through this process, is these long introns. So I told you the exons, those go and create proteins. These introns actually play a role in the cutting up or cutting out of those introns to make different transcripts. And so you can see that different proteins come in and bind on these introns and actually help splice out that section so that you can make an individual transcript. Okay? And if there are mutations in those long chains, you can actually have issues as well. So if you had a mutation in an intron, it could affect how these proteins actually bind here and splice this section out. Okay? So even though oftentimes when we think about disease, we think of, oh, I have a mutation in a gene that codes for a protein. You could have a mutation in a section that doesn't code for a protein, but it influences how these proteins bind to cut out the intron. And therefore, you could have a disease based on that. And it doesn't have anything to do with the actual genetics of that protein. And that sort of blows my mind a little bit, because when we classically think of mutations in the DNA, we think this is a mutation that affects an RNA that affects a protein. But it could affect how that protein is actually created. And so it doesn't have to be a direct mutation to a protein being produced. Okay? So I put a couple other things on here. These little spheres here represent proteins. They also have RNA in them. The whole point of this is for you to understand that RNA is a complex molecule. It's used to code for proteins. It's used for, it has a catalytic activity. It's used for splitting things apart. It's used for interfering. It just is so much bigger than that classic mRNA that we think of. So I've been alluding to this the whole time, this transcriptome. So each one of those little transcripts that I was showing you that codes for a protein, we have a list. There's a list on the NIH website and other websites as well, where you can get all the known transcripts. So every piece of RNA that exists within a human cell or other organisms, there's a list of that. And it's obviously updated because we learn more and more about the cells, human cells and other organisms all the time. So each of those exon those 1, 2, 3, 4, 5, 1, 2, 3, 5, each of those is considered a unique transcript, okay? And we also use the term isoform if it codes for a protein. So we use that term like different isoforms of a protein. And then they also have all these other different types. So if it's a piece of tRNA or rRNA, that transcriptome actually has a list of all of them. And you might say, well, about how many are there? Well, currently there's about 19 to 20,000 transcripts that code for proteins, okay? There's about 50,000 transcripts overall. So there are more non-protein coding transcripts, pieces of RNA in cells or potentially in cells, than there are for proteins. And so either it's a mess in there, that cell, or they actually do something. And I think that as science moves along, we know that they're involved in lots of different things other than just creating proteins. They're involved in regulation and all these different things inside the cell. So the other thing I want you to know is that certain cells, so like I said earlier, my liver cells might be expressing certain proteins and my eye cells might be expressing different ones. And that's a good thing, right? We differentiate our cells. And so a simple study that can be done is to look at the RNA in my liver versus my eye and see that these proteins are being expressed differently. Okay, so there's lots of things that we can learn about humans from these pieces of RNA. So kind of in summary of just this RNA portion is, remember, we have exons that could have mutations, right? That's what we think of simple genetic issues, when you have a mutation in a gene that codes for a protein. But then there's this whole concept of epigenetics. So something that influences how a gene is turned on or turned off. And so those can actually be mutations in introns that cause that. It could be a mutation in a piece of a DNA that codes for a splicing piece of RNA. And so you can see disease doesn't have to be from a mutation in a protein. It can be in a result of, it could be in something that results in upregulation or downregulation or no regulation. And so it's sort of, there's endless opportunities for problems to occur. So it is amazing that we're here having this conversation because of all the potential things that could take place. So so the idea of junk DNA, if that is still in the vernacular of people, is overwhelmingly wrong, because we know that all this DNA is there for a reason. Okay, so where do you get samples? So I keep talking about RNA. There'd be sort of this struggle of how do you take a cell and find out what pieces of RNA are in there? Well, there's a couple different places, and I'm going to talk about patient samples. So let's say you had a patient and you wanted to find out something about a tumor cell versus a regular cell in their body. Or if you took a COVID patient and you looked at their blood chemistry, like the cells in their blood, and you wanted to know what RNA is being expressed in their blood, in those cells, versus a healthy person. And so there are lots of questions that can be asked. And this can be done if you had a lab with a lot of money where you could, and you were partnering with a local healthcare system where you could get samples. You could send out samples and have that RNA extracted. And I'm going to show you an image of that. But there is this national website through the NIH that has all these, hundreds of thousands of samples that you could download and do analysis on. And I did a little bit of that. I actually, you do have to worry about the quality of the data, and it gets better every year. But you can find data to analyze and tell stories about just by going to this website. So those are some of the things that I'll have my students do in classes and in research groups. So this is an important image that sort of shows this process. Okay, so I felt like I needed to tell you a little bit about what that transcriptome was, the RNA that codes for proteins and that RNA that does other things in the cell. So let's say you had a simple situation where you had a tumor, a skin cell tumor. Okay, obviously you could look at normal tissue and you could look at this tumor tissue. Now, one of the things you'd need to do is take those cells and you need to isolate the RNA, because the goal is what is going on with the RNA in this cell and what can it tell us about human health or human disease. So what you do, if you notice, there's interestingly, I like this diagram because it has this poly A tail, and I alluded to that earlier. A piece of processed RNA has a poly A tail. That's what gets it out of the nucleus. Well, you can actually capture all the RNA in a cell using this as one of the techniques you can use. Now, that RNA actually gets turned back into DNA. That's why I mentioned that dogma is complicated, because you can make RNA turn back into DNA, and then kind of magic takes place, and we do all these, we cut these up, we add linkers, and it can get sequenced. Okay, and it's really randomized, and so you have these hundreds of thousands of little pieces of DNA that go back and represent this RNA that was in the cell. And that's where the work that I did comes in. It's how do you take this RNA, this RNA that represents the cells, and what do you do with it? So how do you take all these little pieces of RNA that's been sequenced and make some sense out of it? And I'm going to get into that. But you can see there's all these little pieces because that gets chopped up, and somehow you have to align it back to the transcriptome or the transcripts. And so you have these little short reads of RNA that ultimately go back to what's in this cell, and then we can align them to the known transcriptome. So the known pieces of RNA that exist. Okay, so how do you get that? Well, you can purchase kits. So if you had a cell or a sample of cells, tissue, you can purchase kits that help you break open those cells, get all the RNA. You can capture it with those poly A tails, and then you can amplify it. We have equipment, actually, even in this classroom, where you can amplify DNA, because you turn that RNA back into DNA, and then you can do some analysis on that. And I want you to think about this for a second. If DNA becomes RNA, becomes proteins, you can imagine that certain cells would have genes turned on to code for certain proteins. So even though we know every cell in the human body has a complete set of DNA, not all that is turned on. So I keep using this same example. I don't want my eyes to have the genes my liver needs turned on. So you can, even going back to this image here, if there's a lot of a specific piece of RNA, you're gonna get more reads that exists. So, and that's really important to understand. So you could have a disease where you have a mutation in part of your DNA that makes a specific gene turned on or turned off, or how much it's turned on or turned off. And that's something that we could analyze, because we could assume that if you have a bunch of this one transcript, there's more of it. When it gets through this whole process, we'd have more reads of that one specific transcript. So we'd have more of these little pieces for a specific transcript if there was a lot of it in the cell, okay? Or there might not be any of it in the cell, and we obviously wouldn't map anything back to that transcriptome, okay? So you can isolate and amplify these things. And I'm not going to show this video, but it is amazing how this process gets done. So we cut up all those pieces of RNA and amplify them, and then there's a technique called Illumina sequencing where it generates this huge amount of data that basically tells us how much of those little pieces of RNA we have in our sample, okay? And I encourage you to go to this website and watch this video. It's actually pretty slick to see how fast and how it's actually what makes this technique a potential diagnostic tool. Without it, it would just take too long. So this is actually a pretty fascinating technique. All right, so I've zoomed in on this slide because I want you to see that these little pieces, these fragments of RNA that were determined through that Illumina processing, they map back to a transcript, okay? And a transcript is codes for a specific piece of RNA that we know. Remember, the known transcript. Now, why we're interested in this is we can find out for a specific cell, we can say, well, which transcripts are being created, okay? And we can ask the question, how many of those transcripts are present? And so that feels like a simple thing to do, but it's not. And that's really what I worked on, is how do you take this raw data that you get, these little pieces, and map them back to what is actually happening in a cell? And so there are programs the free programs to do this work. So you take all these files that have this information and you map them back to the transcript, okay? Now, why would you want to do that? Well, it's going to tell you if you have a normal cell versus a tumor cell, it's going to tell you what genes are involved or what genes are turned on in that cell that may be a result of either the cause or some symptom of having that specific tumor. And then you could potentially even look at cells that are precancerous and find out, well, what are those cells expressing gene-wise in here? And so it just allows us to know which pieces of RNA are being created in this cell. And remember, we're mapping back. When you map with the transcriptome, you're mapping back to these particular sequences. So this might be, you remember, this exon and this exon and this exon are connected. That's what that transcript represents. It doesn't represent the whole genome, just that one transcript. That's important, and you'll see why here. And so what we can find out is, are there different exon combinations in a cell that has cancer versus non-cancer? Or one study is you might look at people or children that are, which are people, that are actually exposed to something like particulate matter. So we know that like what we're exposed to can influence which transcripts are being produced. And so human health is influenced by the things around us. So maybe you have an exposure to alcohol in the womb, or you live by a highway where there's a bunch of particulate matter in the air. That can actually influence which transcripts are being produced. And so we learn a lot about how our environment influences human health. We can through this process, okay? So again, those are some things that we learn from doing this technique. And one of the ways we represent these, this is a couple tables I created using different programs, but this is actually what's called a volcano plot. And what it does is it looks at which genes are being overexpressed or underexpressed in comparison to a control. So you might look at a healthy patient versus a patient with COVID, and these can represent different transcript names. And so that's really important when you're trying to figure out what is actually going on in these cells from a genetic standpoint. And then we also represent them because it's statistical. We'd say, there's a range in the samples, but the average is way higher in a COVID patient versus a non-COVID patient. And human health is complicated, right? So you're going to get a range in how people respond to disease. And I think we see that now with my example on COVID is 100 people can get it, and 99 of them recover and they're fine, and then one has a bad effect. And so what is it about that person's genetics that are causing them to have a bad response to this? And so that is one of the questions we can answer using, or at least give information about based on this technique, okay? This RNA, knowing what RNA is present. Now, I wanted to talk a little bit about this alignment. Oh, a little bit more about an alignment component that I worked on. So the past alignment was aligning to the transcriptome, okay, looking at the transcript. There's some limitations to that, because if you were to find one of these fragments, where it didn't align to a known transcript, well, I mean, how would you ever find new things if all you were doing was aligning to the known transcriptome, so the known pieces of RNA? So there are other techniques where you can align to the genome. So you take all these little fragments and you align back to this whole piece of DNA, not just the piece of known RNA rearrangement that exists. You can actually go back to the DNA and align it. And one of the reasons you do that is you might find new isoforms, and so new transcripts can be found. You can find, like, if there's a splicing issue, you can find in patients that, because disease can happen because of splicing, you can find like missing exons that in patients. And so it's really statistically complicated to do this, but it can be done. One of the issues is I highlighted this area right here because you can get sequences of RNA that span a splice. So it would give genetic information here and then here, because that piece was cut out. And so a lot of science in these labs focuses on how that piece is actually spliced out. And a newer technique, instead of going through that Illumina processing, is a technique, and this is something I'm interested in, is what's called Nanopore RNA sequencing. And this is done over in a lab here at MSU. And this is really fascinating because it can take raw RNA, these long strands of RNA, and it can actually feed them through a little protein that's on a surface and read the current that gets generated as a result of every base pair going through here. And it's actually a fascinating technique. But it's really error prone. It's got a lot of errors when it comes through, and you can imagine. but this piece of, in this case, DNA can be fed through this pore, and then it generates a voltage going up and down, and from that you can read A, T, C, G, and oddly, you can actually, if one of those A, T, C, Gs are modified, and you may have heard of like DNA methylation or some modification to a base pair, you can actually pick that up in this technique, which is fascinating to me. So this is something I want to learn more about. It's long read nanopore technology, and it's just another way of analyzing the RNA within a cell. I did put a little image here on how this process takes place. It's busy. I'm not going to go over it, but the same type of thing takes place. You get this long piece of RNA with a poly-A tail, and instead of chopping it up and amplifying it, you actually directly read it. And this just looks like a snake pit here, but that DNA gets fed through, and you can see this means nothing to you and me, but a computer can actually figure out what bases are being are in that piece of DNA from that analysis. And I encourage you to look this up at nanopore technology. It's sort of amazing. Okay, it's amazing what we can do as humans. So what are some of my future interests from this? Well, I want to get more involved in this nanopore technology. I know there are ways that you can take stem cells and grow them and expose them to different things like I was saying earlier, diesel particulate matter or ethanol or whatever. And you can actually find out, well, what type of DNA, how is the DNA getting spliced into RNA as a result of being exposed to this? And we'll hopefully make better decisions about air quality and just human health by knowing this information, that it does have a direct influence on how our bodies grow and respond. In this whole process, one of the things I worked on was not just reading the RNA, but looking for mutations in the DNA and RNA. And that's something I focused on near the end of my sabbatical, and I'm going to continue to do that. Because it's bigger than just which transcripts are being created in a human body. You could have a mutation, a classic mutation, that makes you more susceptible to a bad outcome by being exposed to COVID, or more susceptible to getting cancer. And so there are little mutations that can be studied as well, statistically. And then there are genomes that aren't very well characterized, like the zebrafish. It is pretty well characterized, but there's more to be learned, like with every organism. And then this feels very biological, and I am a biochemist, looking at the chemical modifications that influence gene expression as well. So those are other things that I'm interested in. I do want to thank GRCC for allowing me to go on sabbatical. And I know this is complicated. Like, I don't expect I barely follow it. And I spent quite a bit of time, but thanks to Provost Nettle and Dean Vargo and the committee for looking and approving my sabbatical, and Dr. Jeremy Prokop, he spent a ridiculous amount of time with me, and I'm grateful for that. And then Tim Perriard, actually, he made the connection between GRCC and MSU that ultimately got me involved with this. So I'm thankful that he answered that email several years ago. And I will tell you, without having this opportunity, I mean, I've taught here for over 20 years, and it has reinvigorated me to learn more and just to be involved in research in our community, and it's been a really positive experience. So thank you.